Welcome to the deep dive. Today, we're digging into some really exciting research about potentially regenerating eyesight. We're focusing on one specific gene, Prox-1.
That's right. And your mission, if you stick with us, is to get a handle on how this might help treat serious eye conditions, like glaucoma or macular degeneration, and the key science involved.
But without getting totally bogged down in technical stuff.
Exactly. We've been looking through excerpts from a piece titled Retinal Regeneration via Prox-1 Inhibition. It tackles a pretty fundamental puzzle.
Which is?
Well, why can animals like fish regrow damaged bits of their eye, their retina, when we mammals, well, we basically can't?
Oh, okay, yeah, that's a big one. So the problem is, if our retinal neurons get damaged, say, from glaucoma, that's usually it. Permanent vision loss.
It is. Our bodies just don't have that natural repair mechanism switched on, for the most part.
But you mentioned these support cells, Müller glia.
Yes, Müller glia. They're support cells in the retina, but they seem to have this latent ability. In fish, if the retina gets injured, these cells can actually reprogram themselves. They become new neurons.
Wow. So they can regenerate the tissue, but ours just sit there.
Largely, yes. They stay in their support role. It's like that regenerative capacity is dormant or maybe actively suppressed in mammals.
Which brings us to Prox-1, I guess. Is that the suppressor?
Precisely. The research really points to Prox-1 being a key player here. Think of it less like a simple on-off switch, and maybe more like a master controller that maintains the Müller cell's identity as a glial cell.
Keeping it doing its day job, basically.
Yeah, reinforcing that glial identity, making sure it stays a support cell and doesn't, decide to become a neuron.
Okay, so here's the kicker then. The research suggests if you can somehow interfere with Prox-1.
Uh-huh. If you inhibit it, specifically in mice so far, it seems to release the brake on these Müller glia.
You take the foot off the brake, and they start showing potential to regenerate, to become those needed neurons.
That's the idea. They start to show signs of reprogramming towards a neuronal fate, potentially replacing damaged cells.
How are they actually doing that? Inhibiting a gene inside a cell sounds tricky.
It is. You can't just use a standard drug. They're exploring gene therapy approaches. Using a vector, like a harmless virus, to deliver something into the Müller glia.
Deliver what, exactly?
Tiny engineered proteins, things like intrabodies or nanobodies. These are designed to specifically find and block the Prox-1 protein inside the cell, stopping it from working.
Okay, got it. Directly interfering with its function right there in the cell.
Exactly. It's a very targeted approach.
And the implications, if this works and translates to humans, I mean, it's huge. Glaucoma, diabetic retinopathy, AMD.
Absolutely massive potential. We're talking about shifting from just slowing down these diseases to potentially reversing the damage by helping the eye heal itself. Imagine actually restoring lost vision.
It's a completely different paradigm.
It really is. Tapping into the body's own hidden regenerative toolkit.
Okay, but let's pump the brakes slightly. It sounds amazing, but there must be caveats. Big ones, probably.
Oh, absolutely. We have to be realistic. Prox-1 does other things in the body. It's involved in development elsewhere. So just blocking it systemically, not a good idea. Potential for serious off-target effects.
Right. And messing with cell identity. Does that raise concerns about, like, uncontrolled growth, tumors?
That's definitely a theoretical risk that needs careful study. Any regenerative process needs to be precisely controlled. You want regeneration, not proliferation.
So the challenge is making the inhibition safe, targeted only to the right cells in the retina, and probably temporary.
Exactly. Safe, local, and ideally transient. Plus, there are other hurdles. Human Müller glia seem, well, more stubborn, more resistant to reprogramming than mouse cells.
And the human retina is just more complex overall, I imagine.
Much more complex. And then there's the whole long road of safety trials, optimizing the delivery method, getting regulatory approvals. It's a marathon, not a sprint.
Definitely. But even with all those cautions, the core idea feels so significant, manipulating these genetic switches.
It represents a real shift. Understanding these fundamental mechanisms of cell identity and how to potentially guide them towards repair.
So to kind of wrap up the core finding, inhibiting this Prox-1 gene might coax our own retinal support cells into regenerating lost neurons. That offers real hope for future treatments for blindness.
Oh, yes, definitely. But balance with that caution we talked about. The science is promising, the potential is there, but the path to clinical reality is still long and complex.
Okay, so here's a final thought to chew on. If we're figuring out how to nudge a cell's identity in the retina using something like Prox-1, could this whole concept, this idea of manipulating cellular identity, be applied elsewhere? What about other tissues, other degenerative diseases?
That's the bigger question, isn't it? Are there other dormant regenerative programs just waiting for the right signal? It certainly makes you wonder.